Good afternoon, everyone. So today I feel very proud to introduce Professor Dr. Alakesh Bisai. Professor Alakesh Bisai, FICS, FRSC, FNASC, received his PhD from IIT Kanpur under the supervision of Professor Vinod K. Singh. He did his postdoc at University of California, Berkeley, USA. Professor Bisai joined as an assistant professor of chemistry at IISER Bhopal in 2009 and served various positions such as associate professor, professor, and currently serving as professor, Department of Chemical Sciences, IISER Kolkata. His research interests include strategies for structurally interviewing a marine natural product of biological relevance. The professor's research group's total synthesis has been highlighted in Organic Chemistry Portal as the Bisai synthesis of minus 501N 2018 and the Bisai synthesis of lycoramine in 2023. For his contribution to research, he has received the Young Scientist Award by CRSI 2018, DST Fast Track Research Project 2013 to 2016, and the Indian Chemical Society Research Excellence Award in 2021. Bronze Medal CRSI 2020, Stars MOE 2023, Research Grant CDRI Award 2022 for Excellence in Drug Research, SERP Star, etc. Professor Bisai is a Life Fellow of Indian Chemical Society, a Life Fellow of the National Academy of Sciences, and the Life Fellow of Royal Society of Chemistry, etc. He has published his articles in various reputed journals such as Journal of the American Chemical Society, Chemical Communications, Chemical Societies, Organic Letters, Tetrahedron, Synthesis, etc. He has guided 27 PhD students and currently 14 PhD students are under his supervision. Additionally, he has guided 26 BS and the MS students and worked with 65 project assistants. He has secured, sorry, he has secured 15 extramural funding grants with 11 completed and 4 ongoing. So with this short introduction, I'll like to welcome Professor to present his presentation. Thank you, sir. Now the stage is yours.
Okay, can you hear me?
Yes, sir. Yes, sir.
And my slides are visible?
Yes, sir. Sure.
Okay. So at the outset, I would like to thank the chairperson for very detailed, actually, introduction. And also at the outset, I would like to thank Dr. Rasmi for the kind invitation and the entire team. And I must congratulate on this occasion because this came conversion is really pulled up great speaker, great line of speaker. And I'm looking forward to other talk as well. So what I'm going to do is I'm going to talk for about 30 minutes or 35 minutes and followed by I'll leave you maybe for 5 minutes for if there are any questions. And please let me know if I'm crossing the time. Okay. So with that note, I would like to talk about the total synthesis of natural products. And many times people are very scared to say that, oh, I mean, total synthesis is very tough job. It is not actually. And actually, my talk after that, I just want to, end up with a thought that total synthesis actually is very, very, although challenging, but very, very interesting. Why? So that is what we have to now understand. Why natural product synthesis is very, very important. So I'm just showing a slide here. And if you look at these slides, that all approved drugs. So typically, the synthetic chemist target is to do synthesis of some organic molecules of societal benefit. So if I talk about the societal benefit, the drug discovery program comes in the forefront, including the material science people use various organic molecule in different landscape. So if you look at this slide, although it's little old slide, and I'm sure the new, there is no new review. So this is a review article I'm showing you, and it shows that most of the approved drugs, look at, either they are from biological macromolecules, unaltered natural products, botanical drug, Natural product derivative, of course, there are synthetic drug and synthetic drug based on new pharmacophore. So basically, if you look at S and SM, so this is the particular zone to look at, except that all these actually are nothing but, nothing but nature inspired molecule. Like either they are directly natural product, unaltered natural product, or they are mimic of natural product, or they are sourced from the botanical drug or biological macromolecules. So basically, or natural product derivative. This clearly tells us natural product and their derivatives or the phytochemicals, what nature makes are very, very important in the drug discovery. Now the question comes why? The answer rely on the ADME principle for the drug discovery. If you look at that ADME principle, A stand for absorption, and D is a distribution, M is metabolism, and E is excretion, and of course T is a toxicological effect. So basically, if you think about this ADME principle, where metabolism is very, very crucial part, and where actually drug discovery program face the challenges. Now, what I mean by saying that, so a drug molecule is nothing but a sort of catalyst. If I take a foreign molecule in my, human body, then a drug molecule should go to the system and it should interact with the receptor. So again, that is where you will see that homo lumo, this guiding principle is very, very important in the context of drug discovery. A small molecule goes to biological system, it should interact with the receptor, it should do its job. Of course, distribution is very important. That is why the, this solubility comes into the picture. So if you look at all these, what a ideal drug should be is nothing but a foreign molecule going to the biological system, and it should do its job, and then followed by it should be removed from the body as soon as possible. However, when it's come to, and that is where the beauty comes for natural products and derivative. Why? Because nature knows the best. She knows how to make a molecule and how to break a molecule. If you think about glucose synthesis and degradation, or if you think about fatty acid biosynthesis and its degradation, you will see that nature follows the best practice. What is that? Is nothing but you know principle of nature follows principle of microscopic reversibility. So what it does basically, while making a number of natural product, it can follow a similar path, and that leads to a number of organic molecule. Now what are those molecule should be acts as a good drug? The molecule, after taking, if they follow a circular chemistry in our biological system. So basically this metabolism is very, very important, and that is why it is believed that natural products and this derivative should actually um, are the probably the best drug in years to come. So if you look at the literature, already more than 60% molecules which we consume as a drug, they are either natural products or they are derivative. Now, with that said, we works on number of alkaloids, naturally occurring alkaloids. I'm showing few of them. Like if you look at chimonanthin and folic alicanthin. So now this heteroatom plays actually crucial role, because a drug molecule is nothing but goes to human bodies and go interact with the receptor. And that is where I'm saying that homo lumo principle is very important. Now, if you have drug having heteroatom, they are having unshared pair of electron, therefore they should bind with the receptor. So basically your drug molecule is acting as a homo partner, is going to interact with the LUMO. So now with that said, if you look at the chimonanthin, so this is a complex natural product and having 4 nitrogen atom, and if you look at these structures, it can actually rearranges in presence of acid, leading to another complex structure called calicantidin. Now, how the nature is forming this? If you can break this particular bond, you can think about a radical over benzylic position over there or there. Now, this radical combination can lead to this vicinal all carbon quaternary stereogenic center present in this molecule. Now, how to get this radical? That radical will come from this, tryptamine can undergo oxidative cyclization. Now, if you look at idiospermulin, in idiospermulin, this partner is already there. This is nothing but chimonanthin type of skeleton. But then there will be another hexahydropyrroloindolin that is linked with this aromatic position. Okay. And if you go further complex molecule like Win 64821 or ditryptophenolin and SF5280,451, these are the natural product also isolated, and they how nature is making them. Nature is making from a diketopiperazine prepared from L-tryptophan and phenylalanine. Now, if you have a diketopiperazine monomer, can you do oxidative dimerization? So when nature does, it also gives a pair of diastereomers. And in fact, Win 464821 and ditryptophenolin are nothing but a pair of diastereomers. So nature makes them very easy way. But when it comes to the synthesis in the laboratory, it suffer various problem like chemoselectivity, site selectivity, diastereoselectivity, and also there are a lot of oxidation product that also might lead to a number of undesired products. So there comes the job of synthetic chemist. Can we selectively form a carbon-carbon bond? Now these are other cyclotryptamine alkaloid. So chimonanthin, calicanthin. Now look at Hodgkinsin and quadrigemin. In Hodgkinsin, there are three hexahydro partners are linked. But if you look at quadrigemin C, there you can see four different hexahydropyrroloindolins are linked together. Now, cytolin is the one of the most complex structure in this series. What happened in the presence of acid, this particular junction can undergo, rearrangement that lead to the formation of cytolin. So if you look at the literature, so basically they have been used as traditional Chinese medicine for the treatment of cold, antihypertension, or they are having other biological properties. Okay, so therefore we have been attracted for their total synthesis. There are other alkaloid we also work is called galanthamine. Now, if you look at the marketed drug for Alzheimer's, there are three drugs commercially available. One of them is galantamine, another one is rivarstigmine, and the third one is donepezil. Now, if you look at galantamine, structure, this is a direct natural product isolated from nature. Very recently, if you look at narsipavlin and narsicagenin, just a minute, I just take a few seconds. So narsipavlin and cagenins, now if you look at this particular natural product, this galanthamine is attached with this kind of molecule called galant indole. Now this galant indole is actually can show now pair of atropodiastereomers. So these are the natural product have been isolated very recently in 2021, and we thought of developing a common strategy for galantamine as well as narsipavlin. So if you look at narsicagenins and narsipavlins, so narsicagenin A and B, you can see the difference here. So in these two molecules, there is only one changes. Look at this atropisomerism, this biaryl. So in this case, and this leading to a pair of atropodiastereomers. So basically, this gives atropoisomer, But then if it connect with, this kind of, galanthamine motif, they can lead to a pair of diastereomers. Now, narsipavlin A and B, again, nothing but a pair of atropodiastereomer. They are differ only the oxidation pattern of this one. In this case, it is indolene, but in this case, this is indole. Now, what we did, we actually make this kind of tricyclic core in enantiomer-enriched form. So what we did, we just follow catalytic asymmetric reduction, Ore-Bakshi-Shibata reduction, and in 4 steps, we could actually get this kind of tricyclic motif. Now, this was further elaborated to the formation of dialdehyde, because what we thought, can we have racemic benzylamine, which is nothing but galanthamine, galanth indolylamine, okay, which is also a natural product. So basically what we did, we made racemic form of this natural product, and that we couple with this dialdehyde. So what is happening here? If you look at these two aldehydes, one of these aldehyde is aliphatic aldehyde and another is aromatic aldehyde. Now this aromatic aldehyde is having para-position methoxy group. So therefore, this aldehyde will be less reactive than the aliphatic aldehyde. So what is going on? This is doing first reductive amination with this aliphatic aldehyde, and then it is doing second reductive amination. And by doing that, what we could show for the first time, just last year we have published this work. For the first time, we could show that a double reductive amination of these two quaternary would essentially give us a pair of atropodiastereomer, and from where the first total synthesis of narsipavlins A and B have been achieved. In fact, what we could do is that once this TBS group was removed, that gives us X-ray, and which actually showed that this is the correct atropodiasteromer that we have made. And so this is how we have confirmed its final structure. And I'm very happy to share you that this have been, highlighted by, the chief editor, Jack's chief editor, Professor Eric Carrera, showed that the synthesis commences with the Mitsunobu reaction of phenol, alcohol, and followed by a Hague construct, the central dihydrofuran ring. After functional group adjustment, dialdehyde is coupled with amine G through a double reductive amination. So this actually gives us very, very, solid strategy, because when it comes to drug discovery, you have to have more compound in your hand. Otherwise, all this biological profile cannot be done. Now, coming to my second part, the second part is indolosesquiterpene alkaloids. So now if you look at indolosesquiterpene alkaloids like geamicins A and F and oridamycin A, look at only these two molecules first. So if I look at these two, molecule, they are nothing but diastereomer relationship to each other. Because in this molecule, this particular stereogenic center, methyl group is above and ester is below the plane, whereas here methyl group is below the plane and acid group is above the plane. So one of them called geamicin A and another one called oridamycin A, and they have been isolated way back in 2012. Okay, and they have been isolated from Streptomyces species, and which is nothing but an endophyte from the mangrove plant. Okay, um, if you look at the biological activity, they actually exhibit selective anti-HIV activity. Not only that, there are a number of other congeners have been isolated, like geamicin C, where you can see only benzylic oxidation of geamicin A. A selective, functionalization over there could make geamicin C and D. Now, if you do overoxidation of this benzylic alcohol, that can give rise to benzylic ketone. So basically, if you have geamicin A, they can be selectively oxidized or further oxidized to give the ketone. Now, a recent study also shows in 2021 and 2022, it shows that they exhibit activity against RNA synthesis protein NSP10, which is a major activator for SARS-CoV-2 replication. So you can clearly see They have huge potential in terms of biological activities. They are antiviral agent. Even they have been hypothesized to be used for COVID drug. Now, more importantly, what happened if you look at the monomer? This monomer can now do oxidation. Now, if you look at the monomer, this was the monomer where you have NH. Now, this NH can oxidize to form NN bond. And interestingly, if you look at diamycin A and B, they are nothing but a pair of, again, atropodiastereomer. And these two diastereomers, look at this junction, NN bond leading to a atropodiastereomer. So, interestingly, what is happening once the formation of NN bond happen, this is actually do the restricted rotation because of this perihydrogen, and therefore a pair of atropodiastereomer like diamycins A and B have been isolated in 2012. And it is amazing to show that NN dimer shows 10 times better activities than their monomer siblings. So therefore, there is an interest how to make this NN bond. Not only that, if this monomer oxidized to form CN bond, that is nothing but diamycin C. So here again the oxidation is happening with the nitrogen center with this carbon center, that leads to CN bond formation. Now, since there is no sterics group, diamycin C doesn't show any atropodiastereomers. So in this regard, I would like to draw your attention by one of the great method discovered by Phil Barron from Scripps Research Institute. What Barron group could do, they could take the geomycin A monomer and they introduce the electrochemical oxidation to form NN bond. Okay. So now what is going on here? So basically a single electron oxidation of geomycin A could lead to a radical cation. Now that radical cation can remove H plus and then form this N-center radical. Now this N-center radical can form NN bond. Now interestingly what they could see that in under that condition they could achieve only diamycin B and there was no diamycin A. So interestingly, what is happening here, when you do electrochemical oxidation, you have to use an electrolyte. Those electrolytes could be salt like tetraethyl ammonium bromide. And then what they could see that 17% yield of haloarine is bromoarine is forming under that condition. So this gives the hint that one can utilize just simple bromides, salt like bromides. Bromine is very toxic, anazole is very reactive, and if you look at NBS also generates bromine in situ. So this tetraethyl ammonium bromide could generate bromine in situ under oxidation, and that could undergo an electrophilic bromination. So here we ask the question: why they are only selectively getting diamycin B and why not A? So that is how we came into this picture, and I'm just showing the literature of monomer synthesis of geomycin A. So it is Phil Barron in 2012 has synthesized the first total synthesis of geomycin, and later in 2015, Angley and co-workers showed the 6-pi electrocyclization strategy, oxidative 6-pi electrocyclization strategy to form this aromatic ring. So Professor Richmond Sarpong from University of California, Berkeley also shown a divergent strategy to geomycin and uridomycin starting from carbon. From our country, Professor Dattatreya Dethe has also shown a ligand strategy starting from Whelan-Misser ketone and which can be achieved from organocatalytic, Michael followed by aldol, that is nothing but Robinson-Allinson reaction, starting from 2-methyl 1,3-cyclohexane dione. Along this line, we have been, developing a number of strategy, and few of this strategy we have already reported past few years. I'll be discussing one of them. So what we did, we just made this pentacyclic core. If you look at this pentacyclic core, carbazole is already anchored in this molecule. Now there is, transdecalin, but this transdecalin is functionalized through benzylic oxidation followed by sagucite oxidation leading to enone. Now, if you look at this particular molecule, here there are number of delta position. Like this angular methyl is having a hydrogen that we designated as delta 1. This is a delta 2 position. Okay, starting from this enone, you can just calculate this alpha, beta, gamma, delta. Here is another delta, here delta 3, and here is delta 4. So delta 1, delta 2, delta 3, and delta 4. So the question was, can we functionalize either delta 3 or delta 2? Because if we can functionalize, that could give us the functionalized skeleton for the synthesis of bleomycin and rhodamycin. And here, Dr. Mintu Munda and Dr. Vipin Gavid played a crucial role. So what they could do, they could just take this molecule and then treat with catalytic H2SO4 in acetic anhydride and treated with stoichiometric amount of N-bromosuccinamide. And what they could achieve is, they could achieve the stereoselective formation of this secondary bromide, which is guided by this kind of enone, and which is giving rise to only one particular stereoisomer of this secondary bromide. Now, how exactly this reaction is happening? So we wanted to do some control experiment. So what we did, we just did among various control experiment, we just took this pentacyclic core and then treated with catalytic H2SO4 in acetic anhydride. And what we could see that we could achieve, we can actually isolate 35% yield of this kind of pentacyclic core, and where we could see that this angular methyl is now shifting from this position to the beta position. And when it does this shifting, it is giving a single diastereomers. Now, with this in hand, when we treated with NBS, this compound, this enol acetate, actually gives rise to the formation of secondary bromide, and where again this methyl group is actually shifting from this position to a position. So this is like Wagner-Meruin type shift. If you look at the biosynthesis of complex terpenes, nature does this kind of one-two shift. And this kind of one-two shift, when it happens, that actually always give scene to scene fashion. If it is above the plane, this will shift to the above the plane. Now, what is the proposal, mechanistic proposal? In the presence of H plus, this could probably pull the electron, thereby generating a carbocation, 3-degree carbocation over there. Now, this carbocation will trigger this 1-2 migration of methyl group, and now this is leading to another 3-degree carbocation. Now you look at here, there is a carbazole ring I have not drawn. Now this is situated at the pseudo-benzylic position. Therefore, it could undergo a beta elimination, leading to this a double bond at the benzylic position, which now could undergo bromonium ion formation from the convex phase, therefore leading to so the molecule is like bend size, it's like bowl shape. Now bromonium ion formation is happening from the less hindered convex phase, which is nothing but seen as methyl group. Now what happens, since it is, the bromonium ion, it will try to stabilize by the carbazole nitrogen of this aromatic ring, therefore generating this carbocation, which is more stable carbocation situated at the pseudo-benzylic position of carbazole. Now methyl group again migrate, again 1-2 fashion, leading to this particular compound, where the secondary bromo is getting 100% one of this stereoisomer. So with this secondary bromide in hand, our job was rather very easy. What we did, we just now follow that E2 elimination in the presence of BBU under elevated temperature. Now, with this particular olefin in hand, we did allylic oxidation using SiO2 under heating, and then this kind of dienone further elaborated to the congeners of geomycin. How we did? We could actually chemoselectively reduce this more exposed double bond, and we could get very high yield of this product. Now we can do overhydrogenation of this enone that could again set this hydrogenation coming from the below plane, leading to again the trans-decalin stereogenic center fixed. Now with this in hand, we followed a highly diastereoselective reduction of ketone, and that gives us this greater than 20:1 diastereomeric ratio of this kind of diol compound. Now this in hand, we just did detosylation using solvated electron process. That is nothing but we just added magnesium turnings in methanol, and that leads to the deprotection of tosyl group. And tosyl group is actually deprotecting as a aryl sulfinate. Okay, so overall reduction of sulfonyl group is happening, and deprotection of carbazole is actually give rise to the first total synthesis of geomycin D. Now, with that geomycin in hand, what we went ahead for the formation of NN bond, because in the literature there is only digeomycin B is reported. So our target was to look for the both the arthropod ionomer as prepared by nature. So what we did, we could see that this secondary hydroxyl group was required to protected with the TMS ether, because this TMS ether could be hydrolyzed very easily. Now, in the presence of copper 1 and N-methyl imidazole as a ligands, we could do this aerobic oxidation, and that could lead to the formation of NN bond in the presence of oxygen atmosphere. Then what we did, we could actually do, desilylation, and that leads to the formation of digeomycin A methyl ester and digeomycin B methyl ester. And what we could see that both these arthropod diastereomers can be obtained through oxidation, and since they are arthropod diastereomer, these compounds can be separated through column chromatography. Have this in hand, we did the saponification reaction, and we could see that the saponification reaction was not very simple, probably because of they are situated at the all carbon quaternary stereogenic center. Therefore, if special type of condition like take dioxin and aqueous KOH and LiOH, this is 1:1 KOH LiOH under elevative temperature, we could get first total synthesis of digeomycin A, and this is the second total synthesis after Baron group. Okay, this we have completed in the year 2023. Now, I'll just take 5 more minutes and then complete my talk. Then once we have this kind of monomer, we thought of utilizing visible light promoted NN bond formation, because we have used copper mediated process, but then visible light mediated process are welcome, because in this process the catalyst you have to use actually really low quantity. And in fact, what we could do here is that we could use 0.05 mole percent of iridium 3, and that could undergo in the presence of blue LED, could undergo the formation of NN bond, okay, in the presence of oxygen as an oxidant. That could again give rise to the digeomycin A and digeomycin B. So this is the second synthesis. Just this year we have actually reported. So we have actually unified this synthesis, because the ultimate target is you have to make more amount for the drug discovery program. Now, if you look at digeomycin C, that is nothing but a CN bond formation starting from this nitrogen and the para position of this nitrogen. Now that they can lead to digeomycin C. But another thing which is not yet isolated is the CC dimer, because once you form this C center radical, they can also undergo homodimerization, and that should lead to digeomycin, CC dimer of digeomycin, which is not yet isolated in the literature. However, we believe that the CC dimer is the thermodynamically more stable than the CN or NN bond forming product. So what we did, we just tried to make this compound, and we have seen that that hypervalent iodine can play a crucial role here. And we could see that in the presence of stoichiometric amount of BF3 etherate and hypervalent iodine, we could get the CC bond forming reaction in synthetically useful yield. Okay, this we have published last year. So basically what we could see that we can form NN bond, CN bond, and CC bond. Okay, so these are all leading to a number of dimeric natural product of geomycin. Along the similar line, we have also showed this iridium 3 photoredox catalysis for the total synthesis of digeamycin C. In this case, you need to prepare this CN bond. And what we could do is that although protecting group is very much required for such coupling reaction, but for the first time we have shown that sulfonyl protected carbazole with this bromoarine and the acetate protected of the secondary alcohol would undergo a CN bond formation with geomycin A methyl ester, and where we could see that this secondary hydroxyl was not required to protect, and under this condition we could actually get the CN bond forming reaction in 63% yield. And with this in hand, we have shown this is the second total synthesis, because the first total synthesis was made by Professor Ang Lee from Shanghai Institute of Organic Chemistry Institute. So we are also happy to say that this has been dedicated for, this has been published as a part of CRSI 25th anniversary, which came as a cover page. So this is where now the literature. So basically, geomycin A, we have developed a strategy in 2022. Later in 2024, we have developed another strategy. And so far as this digeamycin A is concerned, we could show two different strategy. One reported in 23, another in 2025. Whereas the digeamycin B, the first total synthesis was done by Phil Warren in 2014, and later we have shown two different strategies in 2023 and 25. We could also show carbodigeamycin synthesis, because we consider that the dimeric products are thermodynamically more stable than the corresponding CN or NN product. Also in this direction, we have shown the total synthesis of digeamycin C following the photoredox chemistry. So with this, I would like to just show one slide, last slide, which I am not going to discuss. So just wanted to show a natural product called silagindorphones A and B. So this has been isolated in 2018. And if you look at this particular heptacyclic core natural product, it is having a spiro center. And now, if you look at this particular site, there are carbotricyclic core. This side also you can see carbotricyclic core. But then here a spiro stereogenic center with a double bond. So basically, if you look at cyclohexene, one thing comes to our mind is to follow a Diels-Alder reaction. And what is silagindorphone B? Silagindorphone B is nothing but, if you reduce this enone reduction of enone, a stereoselective reduction of enone would give the silagindorphone B. And so these have been isolated in 2018. More importantly, they are having some biological activities reported. And for us, the more interesting thing is to how to set this, this 6 stereogenic center in stereoselective fashion. And actually, we have done a Diels-Alder strategy. So basically, if you look at the Diels-Alder reaction, one can think of taking diene that can undergo Diels-Alder reaction with a particular double bond, and that could give rise to the cyclohexadiene. And one can also think that this diene, which is linked with the electron withdrawing group, that can undergoes another Diels-Solder reaction. However, if you look at the normal demand Diels-Solder versus inverse demand Diels-Solder reaction, so normal demand Diels-Solder is energetically much favorable because here diene, electron-rich dienes acts as a diene, electron-rich diene, whereas electron poor double bond acts as a dienophile. So if you look at the scenario, if you combine these two, this can undergo intermolecular Diels-Solder reaction leading to a pair of exo isomer and a pair of endo. But in order to get natural product, one has to get only endo selectivity. Now, if you look at the Diels-Solder reaction of the textbook chemistry, so there the secondary orbital interaction is very nicely, explain why uh, one should get the endoselective product. Okay, so we thought of having similar type of retrosynthetic strategy. We thought of having this kind of die-in and then do a Diels-Solder strategy. And in fact, I'll just summarize my talk. In fact, among all these four possibilities, we could get only endo selective Diels-Solder as exclusive product. And through that, we have shown the first total synthesis actually, first total synthesis of of silagidinorphin B, and we have followed a biomimetic Diels-Solder reaction. And what we could show that this kind of Diels-Solder could gives us a heptocyclic product that actually has been confirmed through X-ray. And Professor Ayan Datta from Indian Association for the Cultivation of Science helped us in doing this DFT calculation of this strategy. So basically, we wanted to make this dinone, but under oxidation, what happened is it is oxidizing. As soon as it is oxidizing, this is giving rise to the LUMO, and that is doing a normal demand Diels-Solder of this kind of diene, probably establishing a cut-in hammer type condition, where as soon as you generate this dienophile, that is immediately reacting with this diene, again following secondary orbital interaction, leading to only endo product, and where Diels-Solder is completely guided by this angular methyl group, leading to the highly diastro selective, highly site selective Diels-Solder and reaction. And not only that, it is doing further oxidation leading to this kind of enone, and which structure was unequivocally proved through the X-ray analysis. So with this, I just want to end up my talk by saying that unified strategy actually help to make not only the natural product but also the various congeners of particular class of natural product. And nature-inspired methodologies are very, very welcome because bio-inspired strategy always help to make compound in more quantities, and asymmetric catalysis also play crucial role in setting up the stereogenic center. And sp3 functionalization makes sometime life easy because this kind of unconventional reaction helps to make advanced intermediate, which is useful for the total synthesis of geomycin A. And we have followed the nature-inspired pericyclic cascade that gives us unique opportunity to make this kind of complex heptocyclic tetraterpenoids. So with this, I would like to thank my institute, IISER Kolkata, and my earlier institute, IISER Bhopal, for the generous funding from the SERC, SERB, MOES, DAE, CSIR, thankfully acknowledge. Also thanks to SERB for the SERB Star Award, and both the director of my institution and all of my students actually who made uh, this contribution. Because of that, I could able to deliver these talks, and I'm also thankful for your kind attention, and I'll be happy to take any question if you have.
Some site.
Thank you for the presentation. Now this presentation is open for discussion. If any audience have any questions, just please ask it.
Yes. Yes, I have Bisai, that apart from this interesting molecular framework and biological activity, means are these atropodiastereomers are showing any special kind of activity so that you are interested to synthesize these things? Means
Yes.
are showing different kinds of activity, but what is the speciality about these atropodiastereoisomers?
Yeah, very, very interesting question actually. The question is, if the monomer is showing biological activity, why one should worry about making the dimer? Number one. And these are atropodiastomers. Why one of the atropodiastomers is better active than the another one? This is a very nice question. Now, so if you look at geomycin A, it is having a number of functionality, but when it comes to the NN bond formation, that leads to the formation of a pair of atropodiastomer. Okay, so now if you look at digamycin A and B, it has been said that digamycin B is having better activity than A. So now the question is why, in how, in which way one of these atropodiastomer is better active than this one. So what do we believe that? So since these are atropodiastomer, so basically if you look about plane, so if you take binol, in that binol case, it is only actrobisomer. But in this case, since the molecule is already having stereogenic center, we call it as atropodiastereomer. Now, if you look at one of the monomer, they are not flat molecule. It slightly bent. Now, if you look at this molecule is doing a dimer, it could be like this or it could be like this. Okay.
Yes.
So now, if this is the case, so you can clearly see that they are having two different spaces, and when it interact with the biological system, okay, they would actually interact with differently. So we are trying to do some theoretical calculation to understand why one of these NN dimers are better active than another one, even why NN dimers are 10 times better active than monomer. This is probably because of the kind of, this special arrangement it is created in the protein. Okay, so we think that this is chirality is playing crucial role in giving the better activities.
Yes, yes. Okay. Thank you, Professor Alakesh Bisai. If anyone else has any question, they can please move forward. And also they can write in our chat box also.
Yeah, Dr. Alakesh, good afternoon. This is Vijay Naganabena after a long time.
Oh, hi, how are you, Jay?
I'm good, good. How are you, sir?
Yeah, yeah.
Very fantastic work. We have been following your work by
Okay,
sitting
thank you.
remote. Very fantastic and excellent work, Alakesh.
Yeah, thank you so much. Yeah.
Just Dr. Alakesh, only one question for you. So this
Yeah.
Daxamine C and all, this rotational and all, how you will see by NMR? I mean, so Daxamine A, Daxamine B.
Very nice question. Yeah, very nice. So basically, if you look at digamycin A and B, they are nothing but atropodiastomers. So therefore, in their NMR, there will be very, very little difference. And one would not able to understand whether really that is different, or if I take several times in the same compound NMR, many times it does some shifting. Okay, so what we did, first of all, they are atropodiastomers. Through column, through some normal column, we separate them. Now, once we separate them, digamycin A and B, we run in HPLC. And what we could see that if I take the mixture and run, I'll see the two peak. Whereas if I take one of these atropodiastomer, it will show the other peak. So basically, through HPLC analysis, C18 HPLC actually, okay, analysis, we could actually confirm that these are two different compounds. And also we have done exhaustive, 2D experiment.